The human brain has 100 billion neurons that control how we think, how we move, and essentially who we are. And for the brain to function as it should, every single neuron needs to be in the right place and making the right connections with all the other neurons. As a neuroanatomist, it's my challenge to understand this incredibly complex structure and how it goes wrong in conditions like Alzheimer's disease. But how do you study an organ that's literally as complicated as the universe? As an anatomist, we have a staple technique called immunohistochemistry. It's a method of labeling tissue using antibodies. We can purchase commercially validated antibodies that bind very specifically to part of the tissue, like a neuron or part of a blood vessel. We then apply a secondary antibody that's attached to a fluorophore, which produces a fluorescent signal that we can see with a microscope. By combining multiple fluorophores of different colors, we can mix and match up to 3 different antibodies to see 3 different components of the tissue. But this doesn't come close to understanding the true complexity of the brain and diseases like Alzheimer's. To do that, we would need to label at least 100 antibodies. And the solution to do that would ideally make use of the resources we have currently available. The vast library of commercially available antibodies that are validated for human brain tissue, and the routine microscopes that are available in many labs. And with collaborators at the National Institutes of Health in Washington, D.C., we managed to achieve this. We took inspiration from other techniques in biology. First, we expanded the number of fluorophores that we could label on the tissue from 3 to 10. And in doing this, we developed a set of custom filters that would work with our microscopes and be able to visualize all 10 fluorophores. We then came up with ways to layer the information on our tissue by adapting an already existing technology to strip antibodies from a tissue and then relabel that tissue. By doing this multiple times, we were able to achieve 10 sets of 10 images from one piece of brain tissue. And the result, I think, is art. It's the ability to label 100 different components of the brain. And it's the ability to see how all of these different components fit together and interact with each other. We're currently coming up with ways to analyze all of this vast amounts of information with machine deep learning. And this technology is making our use of human brain tissue more efficient and more effective. And not only that, but it's taking existing technologies and using it in a way that is going to allow us to truly understand the complexity of the brain and hopefully diseases like Alzheimer's disease.